Hi, this is Dr. Claire. Today we're going to be talking about phylogenies, phylogenetic trees, and how to read them and how to make them. So first let's talk about what we mean by phylogeny. So a phylogeny is simply the evolutionary history of a group of organisms that describes the relationships between those organisms. So which organisms are more closely related to each other, which are more distantly related, when did they diverge, things like that. All right, and then we usually visualize these phylogenies using phylogenetic trees. So this is a simple example of a phylogenetic tree with various branches. Each branch is going to represent a particular organism, and then the branching, how they come together, actually tells you how closely they're related to each other. So let's take a closer look at one of these phylogenetic trees. So this is a phylogenetic tree of the Old World apes. In this tree, time is moving in this direction. So things over on the left side of the image are older. Things on the right side of the image are younger. The other thing that you want to know about these trees is that each of these branching points represents, it's called a node. These represent a divergence event or a timing of a splitting of an organism into two different species. They also represent the common ancestor of those organisms. So, for example, this group right here represents the common ancestor of all Old World apes. Okay? All right. So, the term taxa refers to any group of organisms that we designate. We like to, as humans, group things together and give them names. It helps us to organize things in our brain, and that's just the way we are as humans. So we've been doing this for a long time. We've been looking at things and saying, okay, those things look similar. Let's group them together. So that's a taxon. Okay. Now, ideally, those taxa that we create as humans reflect the evolutionary history of the group. So, a taxa that contains all the descendants of a common ancestor is known as a clade. So, if you're looking at this graph here, you can see that there are numerous clades on here. So, this clade that contains the mouse and the chimpanzee and the common ancestor of those two organisms, we would designate as a clade of mammals. Now, of course, there are more mammals in this tree than are shown in this tree, but just for the sake of simplicity, we're only showing two mammals here. Other clades would be the amniotes, or the organisms that lay egg or have eggs with amniotic fluid, tetrapods, organisms that have four legs, vertebrates, organisms that have a vertebrae, things like that. So we have created these clades, or we don't create the clades, we create the taxa, and the taxa hopefully reflect actual clades. Okay? So if you're looking at one of these trees, and there are two organisms that are most closely related to one another, that's what we would call sister species. So for example, in this tree of the Old World apes, the chimpanzee is a sister species to humans. And they are considered a sister species because they share a recent common ancestor, as they are more closely related to each other than they are related to any other organism in this tree. So that would be a sister species. You can also have sister clades. So in this other group down here, the clade that contains lizards, crocodiles, and pigeons is a sister clade to the clade that contains mice and mice and chimpanzees. Okay, so that's. And you can tell that if you say, okay, well, this is the clade that contains lizards, crocodiles, and pigeons. What's the closest related clade? You go back in time till you get to the common ancestor. And then the sister clade is going to be everything that is downstream of that ancestor. Okay? All right. Okay, so trees also can rotate around any node. It doesn't matter what order you put the things within the tree. They're all exactly the same. So if you're looking at these two trees, they are exactly the same as each other, even though the organisms in the trees are arranged in a different order. And so if you look at this tree, the two sister species that are in this tree, the pine and the rose, are more closely related to each other than to any other organism, and that's the same in both trees. And then pine and rose are then more closely related to fern than they are to moss. They share a common ancestor that is less far back. There's the common ancestor. So they're more closely related to fern than they are to moss. And then finally, moss is the most distantly related within this particular tree. Okay, so it doesn't matter what order they're in, they just rotate. Imagine like one of those mobiles that you would hang over a baby crib where it's got those little branching things and it spins. Phylogenetic trees are just like that. They can spin and it doesn't change the information that's in the tree. All right, so how do we make a phylogenetic tree? Well, you have to decide what you want to look at. So the first thing you need to decide is the group that you're interested in. And there are people who are working on building a tree of life that contains everything in the world in one tree. That's a very big undertaking because there are a lot of things in the world. So usually people are more interested in a smaller subset, so maybe a family of flowers or a order of birds or something like that, and they try to build trees for those particular things. So you have to decide on what you're interested in. That's what we call the in-group. The in-group is all the organisms that you want to figure out the relationships between, okay? And then you have your out-group. Your out-group should be something that's not in your in-group, but it is similar to your in-group. So if you were doing a tree of a particular type of flower, like let's say daisies, which are in the Asteraceae, you would have all these things in the Asteraceae, and then you would have another flower that's closely related to daisies but is not a daisy, and that would be your outgroup, okay? And you have to have that outgroup so you have something to compare your in-group to, okay? So when you're making a tree, you want to look at the traits within the group that are going to help you to cluster them based on their relationships, okay? So generally, you consider that any trait that was present in the ancestor of the whole group, which a lot of times we assume that the outgroup has ancestral type traits. That may or may not be true in the real world, but it's a decent assumption. That those traits are the ancestral traits. So that's what the organism used to look like in evolutionary history. And then you want to look for changes that have arisen within your group of interest. So those would be what we call derived traits. And these are traits that have evolved since the group separated, the in-group separated from the out-group. So in this example here, we have some ancestral traits. They're all vertebrates. They all have 4 legs, things like that. But our turtle is our out-group here. There's our out-group. And so some of the traits that we're looking at within the in-group of mammals are things that have been derived since mammals separated from turtles. So the first thing we have here is hair. All mammals have hair. And so you can lump all mammals based on that trait. Then you might look at something like this, like carnivorous eating teeth. Okay, and some of these mammals have carnivorous teeth. Wolves, leopards, and cats all do. Horses do not. So wolves, leopards, and cats are probably more closely related to each other than they are to a horse. So you group them together in the tree. And then you can have something like retractable claws. And the leopard and the cat both have retractable claws, but the wolf does not. So you put the leopard and the cat together, and that's kind of how you build these trees. Okay? All right. Okay, so let's take a look at one of these sets of traits that you might be interested in. So this is a table of traits from the vertebrates, okay? So these vertebrates have various different traits, and if you look at the outgroup here, the outgroup is the lamprey. The lamprey doesn't technically have a vertebra because it's not made out of bone, it's cartilaginous and it's a little bit weird. But you can look at traits within this group to figure out what animals within the group that we're going to lump together. So you might look at something like mammary glands, which the chimpanzees and the mice both share, and that would indicate to you that those are more closely related. And you might look at keratinous scales, which the crocodile, the pigeon, and the lizard both all have, which indicates that those should be lumped together. Okay, the information about these shared derived traits are also known as synapomorphies, and it's the shared derived traits that you use to construct the tree. Shared derived traits. So just remember that, okay? So it has to be something that's shared between at least 2 members of the group, and something that is derived, meaning it is not the ancestral trait, it is something that has evolved since the group has separated, okay? One thing you have to watch out for in a lot of these trees is what are called convergent traits. These appear a lot when you have convergent evolution. So you have selective pressures that are acting on two different groups in a similar way, and they evolve similar structures. So if you had put in this previous table here, say wings, as a trait that you wanted to group things by, and you had bats and birds in that table, you might lump bats and birds together based on the fact that they both have wings. Now, superficially, they do both have wings, but if you take a closer look at their wings, they are really quite different, and they don't derive from the same evolutionary origin. Bats have solved the wing problem different than birds have. Bats have skin stretched between long hand bones, whereas birds have a flight plane that's made out of feathers. So it's a different way of solving a similar problem, and that's one way you can tell that this is a convergent trait, is because the structure is actually quite different. Okay? All right. So what kind of data can we use to make a tree? Well, traditionally, we use morphology. Morphology is basically just the physical characteristics of organisms, and that's basically what we had when we first started building phylogenetic trees. We only had morphology really to work with. And so that was what was used for many, many years. Things like flower structure, bone structure. You tend to want to use things that are relatively conserved. They don't evolve super fast, because those tend to build better trees, particularly if you're looking at deep evolutionary time. The problem with this, though, is that some taxa don't show a lot of differences, and it's also kind of difficult to compare things that are really different because they show too many morphological differences. So that can be a little bit tricky. Also, sometimes differences in morphology are caused by the environment, not by genetics. So if you grow a plant in a dry environment versus a wet environment, the plant that grows in the wet environment might be bigger and have bigger flowers than the plant that grows in the dry environment, even though they're the same species. Okay? You can also use development. So similarities in the developmental patterns. This actually is good for comparing things that are very distantly related, because embryonic development actually changes very, very slowly. So for example, these little pictures I have here are two things within the Chordata, a group of organisms that include the vertebrates. And at this stage, in the larval stage, these two organisms, the larval stage is basically the embryonic stage, look very similar, right? They both have a neural tube and a notochord, and they're remarkably similar looking. But when they're adults, they look like this. A sea squirt's kind of a hollow bag filter feeder that sits on the bottom. It doesn't move, it doesn't have eyes, and it does not look anything like a frog. But the embryonic development reveals that these are actually more closely related than you might think. Of course, you can also use fossils. We do have a lot of really good fossil evidence for some groups that can show us what the common ancestor may have actually looked like of particular groups. But the fossil record is quite fragmentary, and some things don't fossilize well. So, for example, think about jellyfish. Jellyfish are big and squishy, right? They do not fossilize very well. It's very hard to find a fossil jellyfish, so you can't really use the fossil record for something like a jellyfish. More recently, people have used molecular data to build these phylogenetic trees, and this is actually a really great tool to use, because some parts of the genome evolve rapidly, other parts evolve slowly, so you can use different parts of the genome to get at different relationships at various times in history, and it's one of the most powerful mechanisms that we have for building these phylogenetic trees. So these days it's really commonly used to build phylogenetic trees. And with these with the DNA, basically what you do is you just sequence big chunks of the DNA, and then you look for differences in the base pairs. So for example, in this example here, these two species share a G where everybody else has a C, so that might indicate that species C and D are closely related, so you can put them together on the tree. These four species all share a T, so that's what allows you to put them together, and you can do things like that to work out the relationships between these groups. Okay? All right, so how do these phylogenetic trees relate to the taxa that we make up as humans? Well, hopefully you guys remember your biological classifications. So we have the domains. There are 3 domains. That's the largest classification, and then we have progressively smaller nested classifications within that. So we have domain, kingdom, phylum, class, order, family, genus, species. All right. The way I remember that, I was taught when I was in high school, King Philip chases only friendly Girl Scouts. That's the mnemonic that I remember. Because they added domain since I was in high school, I had to put a dude at the beginning of that. So it's dude, King Philip chases only friendly Girl Scouts. Other ones that you might have heard, kings play chess on fine grained sand, or King Philip came over for good soup. Whatever works for you, that's great if it helps you remember the order of those different taxonomic groups. So this here is an example. This is my favorite bird. This is a red-backed fairy wren, but it is classified within the domain Eukaryota, in the kingdom Animalia, in the phylum Chordata, in the class Aves, in the order Passeriformes, in the family Meluridae, in the genus Melurus, and in the species Melanocephalus. So each organism that you have on the planet, you can classify into all these different groups. Now, in a perfect world, each one of these groups will correspond to the evolutionary history of the organism. So all birds in the genus Melurus, there are about 8 of them, descended from a common ancestor that was part of the family Meluridae, which descended from a common ancestor that was part of the order Passeriformes. So that's, in a perfect world, how it would work. However, we're not always great at naming things. So there are various different types of taxa that we can create. In a perfect world, we aim to create what are called monophyletic taxa. A monophyletic taxa is a group that humans designate, and that group contains the common ancestor of the group and all descendants of that group. Okay? There are also some groups that are what are called paraphyletic, and that's when you have the common ancestor and some, but not all descendants. And then there are polyphyletic groups. Those are bad. We try not to do those. And that's when the taxa that you've designated does not include the common ancestor. So let's take a look at what those look like on a tree. On a tree, if you look down here at the bottom, this blue group is a monophyletic group. It contains the common ancestor and all descendants of that common ancestor. That's what we're shooting for. Sometimes we get paraphyletic groups. There's the common ancestor, and this group contains some, but not all, of the descendants. This group out here falls outside what we would call that group, right? So a great example of this is reptiles. If reptiles were monophyletic, birds would be included within reptiles. However, at least historically, most people have not considered birds a reptile. They probably should be considered a reptile. And so they would fall outside of the group even though they share a common ancestor with that group. Okay, so that's a paraphyletic group. A polyphyletic group is when you have organisms that are not really closely related in the same group. So this group here, this yellow group, the common ancestor of this yellow group is way the heck back here, and the descendants of this ancestor include everything in the whole darn tree. So this is not a very good group. It doesn't make evolutionary sense. Okay, so that's how that works. And I think that's it for this lecture. Check back on the other lecture to look at an example of how to use parsimony to evaluate phylogenetic trees.